guys, Miss Cote here, and welcome to this episode on Operons. Now that we've learned how gene expression happens in the cell, we need to look at how these processes are regulated. Just because the cell has the ability to make a protein doesn't mean that it should just make random proteins all the time. In order to be efficient, a cell only needs to make the proteins it needs at the appropriate time. And before we get into the more complicated regulation mechanisms in eukaryotic cells, let's start by taking a look at how our prokaryotic friends regulate their gene expression. So in this episode, we'll review a mechanism that allows bacteria to control gene expression. This is called the Operon model. So let's get started. Let's look at the production of the amino acid tryptophan. The pathway to make tryptophan from a precursor molecule involves 3 steps, each catalyzed by a different enzyme. Way back when we were studying enzymes, we learned that a cell can regulate the production of tryptophan by feedback inhibition. When the cell makes enough of the amino acid, tryptophan can act as an allosteric regulator and inhibit or shut down an enzyme in the pathway. In this case, enzyme 1. However, another way to regulate this pathway is at the gene level. These 3 enzymes are coded for by 5 different genes. It could possibly be more efficient if you can control the production of tryptophan at this level and not waste energy and resources by not making the enzymes in the first place. Wouldn't it be great if there was one switch that would turn the genes on or turn the genes off? Well, bacteria have a switch like that, and that switch is called an operon. Unlike our genes, bacterial genes that are used in the same pathway, like the 5 genes to make tryptophan, are found right next to each other on the circular bacterial chromosome. And remember, at the beginning of every gene is a region called the promoter, a sequence of bases, including the Tata box, that RNA polymerase recognizes and binds to start transcription of the gene. In bacteria, there's another segment of DNA in the promoter called an operator. This is the master switch that determines if the genes will be transcribed or not. So in bacteria, the entire segment of DNA that includes the operator, the promoter, and all of the genes involved in the pathway is called the operon. Let me explain the basics of the structure of the function of the operator using this diagram. You can find the definitions of the vocabulary terms in your book. First, let's familiarize ourselves with what we're looking at. The blue piece represents the beginning of the genes to be transcribed. These can be referred to as structural genes since they'll eventually become a protein. The green portion is the promoter, and the yellow piece represents the operator. The operator can be turned off by a protein called a repressor. When it binds to the operator, it blocks RNA polymerase so it cannot slide down the DNA to transcribe the genes. So where does this repressor come from? Well, since it's a protein, it must be coded for by another gene. And this gene is found somewhere upstream to the left of the operon. So over here on the left is the gene called the regulatory gene, shown in purple, that codes for the repressor protein. Through transcription and translation, the protein product is formed. Now notice at the bottom that the repressor protein can exist in 2 different forms. By itself, this repressor is in an inactive form and will not be able to bind to the operator. However, referring back to our earlier slide of the tryptophan pathway, an excess of tryptophan can shut the pathway down. Here we have an inactive repressor, and when tryptophan is present, this is referred to as a co-repressor, the tryptophan can bind to the repressor and change its shape. Now the repressor protein is in an active form and can bind to the operator, blocking transcription of the genes needed to make more tryptophan. So let's put it all together. Here we have the operon in its entirety. Because this is the operon involved in the production of tryptophan, it's called the trip operon. Normally in bacteria, the trip operon is on, and the genes are transcribed into messenger RNA and then translated into the 5 polypeptides that will eventually become the 3 enzymes required in the pathway to make tryptophan. The repressor is also made, but can't do anything without its co-repressor, tryptophan. When enough tryptophan is made, the excess can bind to the repressor, change its shape so now it can bind to the operator and prevent transcription from happening. An operon like this, one that's always on and can be turned off by the binding of a repressor, is referred to as a repressible operon. Repressible operons are involved in anabolic pathways, and in this example, we're building the amino acid tryptophan. Now let's look at the other type of operon. The other type is one that is usually turned off, which means that the repressor by itself is made in the active form and immediately binds the operator and blocks transcription. The classic example is the lac operon. So let's take a look. The lac operon is referred to as an inducible operon. Lac refers to lactose, and this operon is involved in breaking down lactose into glucose and galactose. If lactose becomes present in the bacteria's environment, then the enzymes needed to break it down need to be made. The high levels of lactose, or more specifically an isomer called allolactose, shown in green, acts as an inducer and binds to the repressor. This causes the repressor to change shape, making it unable to stay bound to the operator. This makes the repressor fall off, and RNA polymerase is able to transcribe the 3 genes involved in this pathway. The messenger RNA is translated into polypeptides, which will become the 3 enzymes needed to catalyze or break down lactose into glucose and galactose. Inducible operons are involved in catabolic pathways. Here we're breaking down a disaccharide into 2 monosaccharides. The regulation of both of these operons, TRP and LAC, are under negative control. The active form of the repressor switches the operon off. So the opposite of a negative regulation would be positive regulation. This is a mechanism to promote or activate transcription. Sticking with the lac operon, let's look at the catabolite activator protein, or CAP for short. As the name says, this protein is used to activate transcription of the lac operon. CAP works in conjunction with another molecule called cyclic AMP, or CAMP for short. If E. coli has a choice, it will choose using glucose as a food source over lactose. So as we saw earlier, if both glucose and lactose are present, the E. coli will be using the glucose. However, the extra lactose acts as an inducer to inactivate the repressor. This allows transcription and translation of the enzymes needed to break down lactose into more glucose for the bacteria to eventually use. But this is happening at normal levels. As you can see, CAP is over here on the left, and with cyclic AMP at low levels, it is inactive. However, if glucose levels drop to what's considered an emergency situation, and we need a lot of enzymes quickly, CAP is activated by the high levels of cyclic AMP in the cell. Now the active CAP binds to the promoter. This allows RNA polymerases to bind faster to the promoter and transcribe many copies of messenger RNA. Positive control usually happens when you need to amplify a response, in this case making many copies of the enzymes to break down lactose as fast as possible to restore glucose levels back to normal. Hopefully this is helpful in explaining what an operon is. Hopefully we'll have time to run through an operon model in class. I think seeing the pieces move and being a hands-on activity will strengthen your understanding of operons. I cannot stress enough that operons are found in bacteria cells, prokaryotes. While there are always exceptions to the rule, when College Board asks about operons, they are expecting an answer that has to do with regulation of prokaryotic gene expression. I will tell you that one of the biggest mistakes students make is to discuss operons and stating that they are in our cells. Our cells have many other ways to regulate gene expression, and that will be tackled in the next episode and in class. So until next time, be a good human.